This is Mrs. Horseman, and today we're going to be talking on the topic of cellular replication. So specifically, we're going to be talking today about mitosis. Here's what I want you to be able to understand or explain after we're done with this lesson. I want you to understand that one parent cell of a somatic cell creates two identical daughter cells, and I know you don't realize or you don't understand what somatic cells and daughter cells mean yet, but I promise you will. I also want you to understand the advantages and disadvantages of sexual and asexual reproduction. And the last thing I want you to understand is that each somatic cell contains the same amount of chromosomes that the original or parent cell had. So let's get started. So how do you think your body grows or repairs cells that are wounded or damaged? All of us are constantly replacing cells in our bodies. So whether you are growing because you haven't reached your full maximum height that your body says you're going to be yet, or because there are cells in your body that are damaged and need to be replaced, or cells that are just really old and it's time for them to go away, and so you need to replace cells that way. All of those cells are going through a process called mitosis. And the way that I think about that is your toe is one of your body, part of your body, right? You can agree with that. Mitosis, so replication of body cells, kind of a silly little thing. Body cells are called somatic cells. So these are all of the cells that are in your body that create that organism. So it could be liver cells, heart cells, lung cells, brain cells, skin cells, cells that make up the eye. If we're thinking about plants, it could be leaf cells or root cells. If we're talking about fungus, it could be some of the hyphae cells that kind of reach into the ground. Any of those are considered body cells. Those are cells that make up the body part of the organism. So it's everything except for sperm cells or egg cells. We'll get to those in a little bit. Somatic cells are diploid, okay? The prefix di means two. So anytime we're talking about di something, we're talking about two of something. So diploid means that there are two pairs of chromosomes. So all somatic cells are diploid. They have two pairs or two sets of chromosomes. Where do you think those chromosomes came from? Hopefully you said your parents. So every cell has a full complement of DNA in it. And half of that DNA come from your mom, half of that DNA came from your dad. And it makes up the unique DNA complements of who you are. So of those chromosomes, though, humans have 46 chromosomes in their cells. 23 of them came from your mom. 23 of those came from your dad. So when we say two sets of chromosomes, that's what we're talking about. So what are chromosomes? We say that word, but what does that even mean? So every cells have chromosomes, and those chromosomes contain DNA on them. Okay, so DNA, if you unraveled it and looked at it as a big spiral staircase, would be one long strand, okay? But when the cell's getting ready to divide, the cell needs to twist those up, and it needs to make it so that it's organized, so that the DNA is just not splitting up all over the place. So in order to do that, it coils up. So that big long DNA strand coils up really tightly into this X shape that you see, okay? So one strand of DNA would be right here, and then this is the replicated DNA strand. So if you look here, this is called a karyotype. It's all of the chromosomes in a human cell, okay? And they're colored so you can see what the pairs are, but these are actual chromosomes, and you can see this one is very similar to this one is very similar to this one, so this is a pair or a set of chromosomes. Like I said, every human cell has 46 chromosomes, okay? And I don't want you to forget, when we're talking about DNA, we've already learned this, but the DNA is the blueprint that controls the characteristics of an organism. It's what tells everything about that organism. It controls everything that's going to happen in those cells. So DNA is really important. So our cells need to take care of it so that when replication is about to occur, it coils it up into those chromosomes. So how do you think your body grows or repairs cells that are wounded or damaged? Okay, the first thing you need to understand is that cells go through a cycle, okay? So think of it like a clock maybe if you want to. Most of a cell's life is spent in a part of the cycle called interphase. And if you think about it, inter means in between, okay? So it's in between cell divisions. So this is the part of the phase, or part of the cell's life, where the cell is going to grow and it's going to carry out all of the normal cell functions. Like we've already talked about, if it's a plant cell, it's going to be going through photosynthesis. All eukaryotic cells will be going through cellular respiration. We'll talk about protein synthesis at some point in the future. We've already talked about DNA replication, like all of those kinds of things. That's all going to happen in interphase. So I said DNA replication also occurs during interphase, and this is when, right before the cell is getting ready to divide, the DNA is going to replicate, because we said that after new cells are created, each cell needs to have identical copies of DNA, right? So before that can happen, the DNA has to replicate. It has to go through DNA replication. So we end up with 2 copies of each DNA strand. So that occurs during interphase as well. After going through interphase, when a cell gets too big, or a cell gets too old, or a cell is damaged beyond repair, it has to be replaced with another copy. In order to do that, a cell has to divide one time, and we divide that division, we separate that division into phases. The very first part is how the nucleus divides, and then the second part is when the cytoplasm divides. So after the cytoplasm divides, we have the cell cycle. It goes all the way through. The cell grows, it does its normal cell things, it prepares for division, the nucleus divides, then the cytoplasm divides, and when we're done with that cytoplasm dividing, you're going to end up with 2 identical daughter cells. And how do we know that they're identical? If you go back and remember, we talked about DNA replication, making 2 identical copies of DNA. Those DNA strands are going to get split up so that each new cell gets an identical exact copy. So to help you picture it, here's our original cell. We call that a parent cell. And then at the end of cell division, you end up with 2 identical daughter cells. So if you see these cells, I know that they're cartoon pictures, but they look identical to each other, and that's really important. Okay, so what is the cell cycle? I mentioned it, but what the heck is it? So I want you to remember this: I, P, MAT, C, and whatever acronym that you want to come up with to help you remember that. But the I stands for interphase, and we already said that interphase includes where it grows, so G stands for growth, so where it grows and does all of its normal cell things. The S stands for synthesis, so what that means is it's making, synthesis means to make, it's making a copy of DNA. So this is where the DNA is replicated, and then this is where the cell completes the preparation for division. Right here, the PMAT part is actually talking about mitosis. That is when the nucleus is literally dividing, and it's broken up into 4 phases, or 4 sections: prophase, metaphase, anaphase, telophase. And I'll show you pictures of what that means in a second. It'll make more sense. Then the very last part of a cell cycle is cytokinesis, and that is when the cytoplasm divides, and that's when you officially have 2 separate identical cells. So I told you before, if you can kind of think about it like a clock, envision it like this. Okay, so here is where we're going to take this cell and it's going to enter into the cell cycle. So here's where the cell's starting to grow and do all of its normal cell things: cellular respiration, protein synthesis, all of those things that cells do. Excuse me. S phase is where the DNA synthesis or DNA replication occurs. And you can see here, look, we have 1 set of chromosomes here. Now we have 1 and a copy. Okay, so this is where the DNA is copied, and that's what that is indicating. G2 is where the cell finishes preparing for division, and then that cell enters into mitosis. And mitosis is that PMAT, prophase, metaphase, anaphase, telophase, and then it goes into cytokinesis, okay? And then it just keeps going on and on and on. So here's what that looks like, okay? I already told you interphase the cell looks normal, okay? That's when it's doing all of its normal things. So you can see here's a cell. Inside of that cell is a nucleus. And inside the nucleus, it looks like a big pot of spaghetti, basically. All of that is the DNA, and that's what it's supposed to look like, because your cell needs access to all of those different genes so it can turn those genes on when it's time for the cell to do that specific part, okay? That's what the cell looks like during interphase. After interphase, though, our cells enter into mitosis when they're ready to divide. So I told you this one talks about prometaphase, so we're going to ignore that part for now. So mitosis is how the nucleus actually divides. Remember I told you a very long time ago that DNA never ever leaves the nucleus. Well, that's true, but in order for the cell to divide and pull the DNA apart, it's going to have to get out of the nucleus. So the cell gets around that by making the nucleus disappear. So during prophase, the very first part of mitosis, when the nucleus divides, the chromosomes condense. So what that means is they coil up into that tight X shape that I already showed you. So the chromosomes condense, and you can actually see them in a microscope. Spindle fibers, and these are these like fibers that come along and they kind of attach to the chromosomes to help pull them apart. So those start to form. And then right here, the nuclear envelope breaks down. So the nucleus is actually going to fall apart into its more simple components. So basically, you're going to have all these pieces of nuclear membrane floating around inside the cell. They kind of move and get out of the way, okay? So this is what, if we're looking at a cell on a microscope, this is what it would look like with specific dyes that make it kind of glow, okay? So you can see here are some spindle fibers starting to form, and those are actually made out of cytoskeleton. And then all of this is the DNA, okay? And it's starting to condense together, okay? At the end of prophase, the chromosomes are going to start moving towards the middle of the cell. They're going to start coming together like this in the center. Metaphase right here, metaphase, and this is how I remember it, metaphase, middle. So all of the chromosomes are going to be lined up in the very middle of the cell, straight down the center. And you can see how these fibers are coming and they're attaching right here. So this says mitotic spindle is fully developed. So all of these spindles have completely attached to the chromosome. The chromosomes are lined up in the metaphase plate, is what they call it, the equator of the cell. And each sister chromatid, so each piece of the X-shaped chromosome is attached to a spindle fiber. And if you look here at this really cool picture, all of these spindle fibers, the green ones, are reaching towards the center, and then all of the DNA, you can see the coiled up DNA is focused right in the middle of that cell, right down here. Okay, so why do we need those spindle fibers, those green things that are coming in and attaching to the chromosomes? Because they're going to play tug of war. So they're going to start pulling back and forth on each other until the chromosome actually splits apart. It's going to be pulled apart in the middle. And then those spindle fibers are going to shorten, 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 shorten as it pulls those chromosomes to the sides or the poles of the cell. So you can see here the sister chromatids, now that they've separated, are called chromosomes again, just to be confusing. Are pulled towards the opposite poles. And you can actually see the cell is starting to elongate. So it's starting to become, instead of a round shape, it's going to become more like football shaped. And you can see right here, this is kind of towards the end of anaphase. The chromosomes have been pulled apart and you've got a whole set of chromosomes over here and a whole set of chromosomes over here. And then the very last one is telophase, or some people call it telophase. And this is where the chromosomes have fully arrived at the opposite poles. They're going to start to uncoil. So they're going to start to unravel from that tight coiled X part shape. The nuclear envelope is actually going to start reforming. So the pieces of that big nucleus are going to move away from the center, and then they're going to reform on the sides where all of the new set of chromosomes are. So the nuclear envelope reappears because we have to keep that DNA safe. And then those spindle fibers are actually going to start to break down. The very last thing, I'm sorry, let's go back here. I didn't show you this picture. You can kind of see how this is like really condensed in that one area, the DNA, that purple stuff. And then right here, it's kind of like spreading out a little bit. It's not as dark purple. It's because it's starting to uncoil. And you can't actually see the nuclear membrane in this picture, but I can tell you it's forming. Okay, So the very last thing, after the nucleus is completely divided and we have two nuclei in the same cell, then the cytoplasm itself has to divide. So this is a really cool picture from a scanning microscope that is showing the cell actually pinching in on itself. To me, it looks like a peach, like a ripe peach that you pick from a tree, but it's actually like pulling in on itself, and eventually it's going to pull all the way in and meet at the center, and then it's going to pull apart, and you're going to have two identical daughter cells. So this, if you're following along with my notes, this diagram is in your notes, and so I want you to pause it here, and I want you to go ahead and label all of the different phases so that you have those names paired up with the pictures. So go ahead and do that, and then we'll get back to the notes. Okay? Okay, so what is the result of mitosis and cytokinesis? Like I said, we have two identical cells. Each of those cells has the exact same amount of DNA. So if I started out with 4 chromosomes, those chromosomes are going to replicate, so now I have 4 sets of chromosomes. So 1, 2, 3, 4, 1, 2, 3, 4, those chromosomes are going to pull apart. So each new cell is going to end up with 4 chromosomes again. Okay, so that's what I mean by identical. And that DNA replicated, it made an identical copy before the cells even divided. So that's how we know that the cell has identical DNA in it. After cytokinesis is complete, so after that cytoplasm completely finishes pinching off, both of those new little cells are going to go back into interphase. So the cell's going to start growing, and it's going to do all of its normal cell things again. Okay, so let's talk. That's mitosis, and that is how your body cells make new ones, in a nutshell. That is a form of asexual reproduction. So without any sexual sex cells coming together, your cells are making new ones, okay? The same kind of thing happens with organisms that are reproducing without the sex cells coming together. So asexual reproduction is actually how prokaryotic cells and some simple eukaryotic organisms reproduce. This can occur very quickly. Some bacteria, most bacteria, can reproduce about every 15 minutes. So if you think about it, on your desk, you have one little itty bitty bacteria, we'll just pretend, one little bitty bacteria. In about 15 minutes, that bacteria has grown and divided, and now there's 2. So in another 15 minutes, those 2 bacteria have grown and divided, and now you have 4. So within a half an hour, you have 4 identical bacteria cells. And it's just going to keep getting more and more and more and more and more. And that's why I say ew, because the thought of bacteria cells growing and then you touch them, and that just grosses me out. Asexual reproduction is when one individual produces offsprings that are genetically identical to itself. They don't need another organism to mate with. They reproduce, and the end result of that reproduction is another identical organism. Just like when we went through mitosis and it made 2 identical daughter cells. This is a little bit more of a simple process because the nucleus doesn't have to divide. There's no nucleus in bacteria. But it replicates the DNA, splits apart, so now you have 2 identical bacteria. Many organisms actually reproduce this way. Bacteria, yeast, which are single-celled eukaryotic organisms. Sea stars can replicate this way. Sea anemones, bees, wasps, and some reptiles can actually reproduce asexually. There's a few different kinds of asexual reproduction: budding, fragmentation, or regeneration. So that's when like a piece falls off and it grows a whole new one. And something called parthenogenesis. So I'm going to show you really quickly some examples. So this is budding. So this is where it's a little hydra, like a little water organism. And that hydra is actually, you can see this little nodule, and that nodule is going to keep getting bigger and bigger, and it actually forms a bud. Okay, and that bud is going to break off. And then once this hydra like fully develops, it's going to be an identical copy. They're going to be completely the same 2 hydra. It's kind of cool because the hydra doesn't have to go anywhere to find a mate. Okay, it just can pull a piece of itself off basically. This is fragmentation or regeneration. So this is a planaria, which is a worm, okay? And if you split that worm into different pieces, as long as it has the nerve cord, like some of the nerve cells with it, then that worm will develop into 3 identical planaria. And then parthenogenesis, I'll show you a video in class. It's actually kind of cool, but parthenogenesis is how some of the more complex organisms reproduce when they are kind of in extreme situations where there are no organisms of the opposite sex. So we'll kind of get into that a little bit later, but it is kind of cool. So we talked about what asexual reproduction is, when organisms reproduce without, sorry about that, when organisms reproduce without coming together. They don't need a mate. So what are some of the advantages of that? Well, if you think about it, organisms that have to remain in one particular place and can't move to find mates, or organisms that are in extreme environments where it's difficult for them to come across another like organism to be able to reproduce, they can make offspring without having to have a mate. So they can continue their species or continue their population. Another advantage is asexual reproduction can occur quickly. It doesn't take long, like I said, 15 minutes for that bacteria to reproduce. So numerous offspring can be produced quickly or without costing the parent a lot of energy or time, okay? And then, like I said, environments that are stable and experience very little changes are the best places for organisms that reproduce asexually. And that's just going to be because they don't adapt very well to their environment because they're all the same. So those are advantages. Let's talk about disadvantages. Because if it was all advantageous, then we'd all just reproduce asexually, and there would be like 3 times as many humans as there are right now on Earth. That doesn't happen though. So here's some disadvantages of asexual reproduction. Asexual reproduction creates an exact copy of that organism. When that happens, there's no genetic variation, okay? If you think about the human population, there is so much genetic variation. Not any one person, unless it's an identical twin, looks just like the other one, okay? And that is a good thing. There's differences within that population. With asexual reproduction, all of the organisms are genetically identical. So if one of them has a weakness in their DNA, they all have the same exact weakness. So if that stable environment changes, then the consequences could be that it wipes out the entire population. Let me give you an example. Bacteria are susceptible to some of the medicines that we take, and what they do is some of the medicines that we take actually poke holes in the cell wall and the cell membrane. And then that causes the cell to die because all of the stuff that's inside the cell leaks out. So if all of the bacteria cells have the same DNA that do that, then all of the bacteria cells are going to be susceptible to that medicine, which is good for us, bad for the bacteria cells. Now, if you have some that have variation, so we have a bacteria cell that when it is exposed to this medicine, it's going to start to break down and it dies, okay? This bacteria cell is not susceptible to that medication. So it has DNA in it that says, you're not breaking down my cell wall, okay? It doesn't respond to those chemicals. This bacteria is going to thrive, and it's going to reproduce and grow and reproduce and grow and reproduce and grow. So that's where no genetic variation is a disadvantage. The more variation there is, the higher chances that even if there's changes in the environment, the population can survive. And we'll get more into that when we talk about evolution. But just so you are aware of genetic variation and why that's a good thing in the population. Okay, so we have come to the end of this lesson, and I just threw a lot of stuff at you. We talked about somatic cells and diploid, what diploid means. We talked about the cell cycle, so interphase, mitosis and all the phases in there, cytokinesis, And then we talked about asexual reproduction and why that can be a good thing, and why asexual reproduction maybe isn't so good either. I really hope that you are able to ask me questions in class, and we'll definitely practice this information. But for now, that's it, and I will see you soon.